Okay, well, welcome everybody. Thanks so much for coming. Happy to have you here. I'm Rob Peters, Dr. Rob Peters from the biology department. I know some of you, but don't know others, so thanks for coming. Are any of you science majors? Oh yeah, what's your major? Okay, awesome, great. And what brings you here? Great. What's your academic background or interest? Okay. Poli-sci. Okay, cool. Okay, great. Well, glad you could join us. So I've structured this talk primarily for a non-scientific audience, figuring there'd be a diversity of backgrounds, but there is certainly science. And I've tried to focus it on sort of more of the bigger picture ideas and discoveries that related to my sabbatical research. So rather than spending a ton of time going in the details of every experiment I did, or I was a part of, talking more about the bigger picture implications. So first, sometimes people start off thanking people and acknowledging them. So I'm going to do that. I kind of like that approach, particularly because this was very much a collaborative effort. So I was in Bozeman, Montana for my sabbatical at Montana State University, and I was co-hosted by Dr. Seth Walk and Dr. Doug Kaminsky. And Seth was a postdoctoral scientist at University of Michigan when I was a graduate student. And I meant to get a picture of him and I from 12 years ago when we were both there, but I didn't get around to putting it in this talk. But anyway, this is Seth and his wife Heather and his child David, and they're walking around Bozeman, and you have the Bridger Mountains in the background, which was a particularly nice setting for a getaway for a semester away. And Heather also works part-time in Doug's lab as a researcher. Basically, she has her PhD in immunology, just like I do. So I worked with Heather, and then very important person here, Carlin Blackwell, and this is her husband Creed. Carlin was a graduate student, and this project was sort of her focus, and I was there to participate and help with the project. So she had been working on this project before I got there and continues this work now that I'm gone, which hopefully increases the likelihood that it will get published at least once. So thanks to those people, and of course, thanks to Aquinas for supporting me and as I went to my sabbatical, and I'll say that again at the end. So a title I came up with here, Helminths and the Hygiene Hypothesis, and Carlin proposed this subtitle, Untangling a Tripartite Symbiosis. So that's a little bit technical for non-science people. Helminths are basically worms. A lot of times they're microscopic, very small worms that infect warm-blooded mammals mostly. They infect people around the world and other animals. And we don't really hear about them a lot in developed countries like the United States because we have a relatively high level of sanitation. So people infrequently get infected with worms. However, in developing countries where they don't have as good a sanitation systems and less public health measures, millions of people around the world are infected with helminths every year, and there is significant disease, morbidity, or mortality caused by them. So that's a little bit of intro about helminths. We'll talk more about them. Then the hygiene hypothesis, I was going to start off by asking you, how many people have heard of this expression from someone other than me, the hygiene hypothesis? One? Only one. Okay, well, that's great. So I kind of like that sometimes when my students say they haven't heard of what I'm going to talk about, because that means I get to teach you something. So great. So I'll get to teach you about the hygiene hypothesis. I'm a little surprised not more people have heard of it, but that's okay. It's been around since about 1989 is sort of the origin. So what's your name, if you don't mind me asking? Ella, do you remember what you heard about it or the context?
Like the whole thing where you get exposed to more kind of like pathogens, and I guess also like other like parasitic organisms and stuff, you'll probably have a stronger immune response like you have the first exposure, so then you have your second exposure or exposure to other like pathogens like allergens or whatever. You'll have like a much faster more efficient
Okay, great. So Ella did a really good job summarizing that. So just to restate, I'd say that's probably consistent with sort of the general understanding, sort of I guess what most lay people would consider the hygiene hypothesis, and that is that a lack of exposure to microbes during our development impacts our immune system in a way that's negative. It either, Ella suggested maybe makes us more susceptible to infection. That's kind of a different idea, but she mentioned allergens, and certainly that's kind of the key here, is that it makes our immune system more likely to respond to allergens, which are things that should be harmless. It also likely makes us more susceptible to developing autoimmune diseases. And I didn't get a lot of autoimmunity stuff in here, but that's a situation where your immune system is attacking yourself and it's not supposed to be. So things like multiple sclerosis, type 1 diabetes are a couple classic examples, lupus. So Ella nailed it, and that is sort of the classic idea. So here's a nice schematic I got from Carlin. It's the exposure to those microbes that Ella mentioned is particularly important early in life. Even during the development of the fetus in the womb, can impact the susceptibility of that later adult. So here we see in one scenario where for many years our species and our ancestors have developed in what you might call a natural environment, where we're outside playing in the dirt, exposed to a broad variety of microbes. We're eating a very different diet that has a lot of plants and fiber. And that's going to really change our microbiome, which is the microbes that live in and on us. That also, as Ella suggested, influences the way your immune system develops. And here we have something called Tregs, which are a type of white blood cell that helps calm your immune system down and makes it less likely to over-respond to things like allergens or to yourself, which could cause autoimmunity. So you see this natural environment, diverse microbiome, helps to suppress extra immune responses and prevent things like allergies and autoimmunity. In today's modern world, in a more unnatural environment, we have reduced diversity of the microbiome, reduced exposure to microbes, which seems to be causing an overreactive immune system and a higher incidence of diseases like allergies and autoimmunity. So that's a summary, and I have a little bit of the history behind this just because I thought it was interesting and kind of gives you a little bit more context. So this was first sort of proposed in 1989 by Strahan. I looked this paper up for this talk, but interestingly, he only ever mentioned the word hygiene in the title and maybe once in the conclusion. He didn't actually coin the expression hygiene hypothesis in this first paper. And all he did in this study was look at basically family size and the incidence of hay fever or eczema. And eczema is sort of an allergy skin condition that involves a hyper responsive immune system. And you can see, I reproduced this from his paper to make the font bigger in case people couldn't see the copy and pasted. But in families where there's a lot of children and a child's born into a family with lots of older siblings, you see a much lower percentage of hay fever and eczema. So 8% hay fever versus in families where a child's born and they're the first child, 20% hay fever. So what's the idea there is, well, he kind of suggested that if you have fewer siblings, you're probably getting fewer childhood infections. And he also mentions that our standards of cleanliness have increased in years. And so our indoor areas have a higher degree of cleanliness. And therefore, you're getting fewer infections and more allergies and what we call atopy, which is a word that means generally susceptible to allergic-like conditions. So this was the first sort of the founding of this hygiene hypothesis. Stop me if you have any questions, I guess. So 1989, obviously a lot of time has passed since then, and there's been a lot of research on this since then. So I'll just give you sort of a couple updates. So in 2002, infect of infections on susceptibility to autoimmune and allergic diseases. This was a review done by Bach in 2002. And this is still what I would call the early days of the hygiene hypothesis. And this is correlation. So make sure you notice this is correlating two different trends. One trend is decreasing infectious disease in recent history, things like rheumatic fever, hepatitis, tuberculosis, mumps. So we see around the world, and especially in developed countries, incidence of these diseases are decreasing dramatically. And at the same time around the world, conditions like here we see an allergic type condition, asthma, and we see a few autoimmune diseases. Crohn's, well, Crohn's is sort of a hybrid between an allergy and an autoimmune disease. Your immune system is responding to the bacteria in your gastrointestinal tract inappropriately. Multiple sclerosis, and type 1 diabetes, classic autoimmune diseases. So this is obviously correlation, but Bach went on to show some other things that suggest that, and this is where the idea takes place that we really have to, what a lot of people think is we need to be getting infected by stuff in order for the immune system to develop. And that idea still kind of, I think, has roots in what a lot of people think of when they hear the hygiene hypothesis, which is why I was interested to hear what you thought about it, Ella. And she did mention, hey, Daniel, she did mention infections. But one of the things I want to do today is make sure it's really clear that many of the experts in the field now suggest that we kind of move away from that idea that hygiene is bad, and that infections are required, or that infections are required for a proper immunodevelopment. So there is strong evidence now to suggest that early childhood infections with common viral and bacterial pathogens does not protect against allergy. So even though we see correlations like this and think, well, we need the kids to be getting sick, that's not necessarily supported. It's more so disruptions in the microbiome, as I kind of referenced before. And so that does kind of tie in the idea of hygiene, but I wanted to use my opportunity here to sort of get the message out there that we still want to practice good hygiene, and we still want to, get childhood vaccinations, because the protection offered by those types of things is important and does not impact the development of those conditions. What things do impact? What does the evidence say more clearly now with 30 years more evidence? What are some of the key points that relate to what we might call the hygiene hypothesis, or might sometimes be called now the disrupted microbiome, or something along those lines? They're still struggling to come up with a good alternative name. But things like the birth route. So if you haven't heard this, when children are born through a cesarean section, the microbes they're exposed to at birth are dramatically different than the microbes that they're exposed to when they pass through the vaginal tract. And that has major implications on the development of allergies and autoimmunity later in life. And I'm not going to go through the data for each of these things. I mentioned diet earlier. Our diets now are much more refined. We eat a lot less fiber, a lot less plants. And in order to support a healthy and diverse microbiome, you need to be feeding them the types of complex plant molecules that they like to produce. And I don't think I'll talk much about this today, but additionally, those microbes produce anti-inflammatory molecules when they digest those complex plant molecules. So like I like to tell my students, feed your microbiome well, and it will treat you well in response with sort of an anti-inflammatory kickback. Animal exposure, this is a big one that I read a lot more about in preparation for this talk, and even has some promise for how we could potentially create some treatments around these ideas. And so a lot of the evidence deals with people that grow up on a farm and people that grow up not in a farm, and we see really striking impact on animal exposure. And why is that? Well, back to this idea of microbiome. If you're playing with animals, and it may sound gross, but you're probably interacting with that animal's feces at some point, whether you see, whether it's microscopic or not, or whether you have pets in the house. Either way, that's going to diversify your microbiome, and both pets and growing up on a farm is associated with decreased rates of allergies, autoimmunity, and asthma. So animal exposure is a big one. Antibiotic use is a big one. That's another one of the things that's increased quite dramatically in the last 50 years. And every time you use antibiotics, you hopefully are treating an infection. And certainly we want to take antibiotics if we have a bacterial infection that we need to get rid of. But you're also disrupting your microbiome. And especially in those early years of life, if you're disrupting your microbiome, you're setting yourself up for increased risk of allergies, asthma, and autoimmunity. And finally, now to get to the other part of my talk, helminths. It is very clear that these worms that prior to modern sanitation were a regular part of our life and development dramatically alter the host immune response. And it makes sense, if they're going to live inside of you, they need to be hiding from your immune system, right? We're trying to kick them out. They want to stay there. So they have a lot of intricate ways to manipulate the host immune response. And lots of evidence shows the way they impact the host immune system makes the host less susceptible to allergies, asthma, and autoimmunity. So helminths are a key part of this hygiene hypothesis as well. So here's some of the data, some of the early data, I guess, around that, just a couple papers. So this is one of the earlier papers from the year 2000, so getting older now. They're looking at children in Eastern Africa because that's a place where those types of infections are still pretty common. And a couple different types, filariasis, and I'm not sure which this one is, hematobium, but these are both helminths, infectious worms. Filariasis is microscopic. You can see here a scale 1 centimeter, so really small worms that infect people and can cause serious disease. But they're looking at whether or not these children were positive for this worm infection. So children that were negative for worm infection, children that were positive for worm infection, and with 2 different worms, children that were negative for worm infection, children that were positive for worm infection. And then they looked at whether or not they had signs of allergies or atopy, that word I mentioned earlier, atopic, which means your immune system is predisposed to over-responding to allergens. And you can see for the children that were positive for worm infection, very low rates of atopy, whereas the children in the same country that were negative for worm infection, much higher rates of allergy. Right. So that's sort of some of the early evidence. And as a result, in the last 20 years, so this is just one little paper. There's lots of research that has gone on to show these sorts of trends, which have really captured the attention of public health officials and scientists and physicians. So there have been a lot of additional attempts in the last 20 years to use these worms to actually treat some of these conditions, allergies, asthma, and autoimmunity. So here are just some snippets of some big papers in the last 20 years that have done this. So Trichuris suis seems to be safe and possibly effective in the treatment of inflammatory bowel disease, an example of an hyperactive immune system. Trichuris suis is one that has been used a lot in the research because it actually infects pigs. And since the pig is the natural host, the infection in humans is self-limiting, which means it's going to end on its own. It's not going to continue to infect you. It's not going to cause a lot of problems. So the good news is using Trichuris suis as a potential therapy, it's not going to spread to other people. It's not going to have side effects. It does require multiple treatments because the infection is relatively short. So there was some promise early on. Pardon me? Okay. There was some promise early on, 2003, that Trichuris suis may be helpful, and it was tested in a variety of diseases. So you see inflammatory bowel disease from this group, Weinstock's group, Crohn's disease, another condition that involves a hyperactive immune response. Here's a different hookworm, Americanus. Oh, I've got to check my notes. Oh man, did I forget to put that in here? Necator americanus, thank you. Necator americanus, so a different worm infection looking in asthma, and you can see they've moved on to randomized placebo-controlled trials. And I'll get to the punchline here in a minute if you're wondering how's this all gone, but basically we haven't cured these diseases. So that's, this Necator americanus is a pathogen of humans, and it burrows into your lungs and, from your bloodstream, and then travels up your trachea, goes into your esophagus, then goes down into your gastrointestinal tract and hooks its mouth into your intestinal tract mucosa and proceeds to suck your blood and live off your bloodstream. Yeah, pretty disgusting. Sorry for, oh, I forgot you're a poet, huh? Sorry. You're not eating lunch, so I thought it'd be okay. But you can see why there might be some hesitation to use this as a treatment, right? And heavy worm burden with this worm can cause anemia and malnutrition. I think I'll mention that again in a minute. But nonetheless, it hasn't stopped people from attempting to use this to treat some of these conditions. So here's another one with Necator americanus and celiac disease, so an allergy to wheat. And this is kind of where we are, I guess, more recent, looking at, well, how recent is it? 2013, so a little more recent than some of these others. They got to a phase 2 clinical trial looking at Trichocereus eggs in treating Crohn's disease, and 2 different phase 2 trials had to be shut down because they were not showing efficacy. So the bottom line is, so far, helminth therapy does show promise, right? We see this, and this is just a handful of some of the studies, but it has failed to deliver in larger clinical trials. Additionally, as I mentioned, helminths can cause significant morbidity and mortality if you're infecting people with them. And so that complicates the use of them therapeutically. So where might we be now? Here's a paper from 2022 that suggests, how could we potentially use molecules that are derived from these worms to potentially intervene in some of these immunological conditions? So interesting prospect, right? So, but in order to do this, obviously, we need to fully understand what molecules are the worms using to manipulate our immune response, and how do those act, and on and on. So an additional complication to understanding this is that helminths manipulate the microbiome. And I haven't said a lot about the microbiome, but it's pretty, I think there's a decent understanding of what the microbiome is in lay population. How many people have heard that expression before? Okay, great. So what's your understanding of it? Does someone want to share their understanding of it? Ella did.
Stuff in your gut, I guess it's skin too, I learned that.
Yeah.
I have a friend who's always drinking stuff that has got microbiome.
You are trying to drink stuff? Good, good. Yeah, yeah. Yeah, Dane, kefir. So yeah, Steve said gut, but also the skin. So microbes that are living all over us, in and on us. Most of the time, I think we hear about bacteria, and that's certainly true, but there are archaeal organisms, there are viruses, there are fungi, there are protists, but usually we're talking about bacteria. So it turns out when the host becomes infected with a helminth, that changes the microbiome, and we also know that changes in the microbiome impact allergies, asthma, and autoimmunity, as I've been talking about today quite a bit, right, with the hygiene hypothesis and much of this research. So you can see sort of the complication here in understanding what's the worm doing if it's changing the microbiome, and the microbiome's changing the host. So I'm going to talk more about that in a minute, but I'll point out that this was work done by my friend Seth when he was a postdoc at the University of Michigan in 2010, published in 2010. So he has some history in working in this field and is sort of getting back into it as one of his several projects. So how might changes in the microbiome be the influence how helminths impact host immunity, which is the question I kind of already posed. And really interesting 2015 paper that sort of illustrates this is here. So I'm going to walk through this and try to get you to participate a little bit here, just because that helps, I think. So one of the worms that I'm going to be talking more about today is Heligmosomoides polygyrus bakeri, which I'll call H. poly, or is referred to as here as HPB. Yeah, impacts airway inflammation in asthmatic mice. So I'm going to, this was a pretty big study. Immunity is a pretty high tier journal in biomedical science. So I'm going to break this down for you, and you can tell me what you see. So HBP is a helminth that we've talked about, one of these worms. And this is infecting mice. This is a worm that infects mice. HDM is house dust mite. So they inject this house dust mite into the noses of the mice, and the mice develop asthma-like condition. Eosinophils are a white blood cell, part of your immune system, that fight parasites like H. poli, but also promote allergies and asthma. And you can see on the y-axis here, they're measuring eosinophils in the lungs of these mice. So audience participation, what do you see? Everybody stop and take a minute to soak that in. Ella's got an answer already, which is great. So Ella, what do you want to tell? Go ahead, tell us. Definitely. Okay, thanks. So Ella's mentioning the star here, which means some statistical tests have demonstrated that this difference is unlikely to be due to chance. So this is likely a real difference that we're seeing. And so does someone want to add to that? Dane? Yeah. And we know those are a cell type associated with a condition like asthma. So yeah, great. Dave said injecting house dust mite or being exposed to house dust mite in the airway, in the nasal cavities, increases eosinophils in the lungs, seems to be promoting asthma-like conditions in these mice. And then?
Your immune response does the same thing. If the white blood cells go to fight the dust mites, but then they also make your asthma.
Well, yeah.
HPV lowers the allergy response. There's less allergy happening.
That's where I wanted you to go. But Steve, can I clarify something for you?
Yeah.
Okay. What's your question?
I just saw that you get the dust, you got a little mouse. Let's get some dust blowing up his nose. All right, gets asthma. Right, then his body, little mouse's body, releases these
eosinophils, yeah.
Right, the white blood cells to fight the dust mites. No?
Well,
that's my
yeah, let me clarify that. So they're doing this exposure 3 times a week for 3 weeks. So they're kind of developing a chronic condition. And so I wouldn't say so much that the eosinophils are fighting the dust mites. I would say that the eosinophils are causing the asthma. Yeah, eosinophils fight parasites, but promote allergies. Because the dust mite is response to the dust. Yeah, the response to the dust mite is an asthma-like condition. And the eosinophils are a sign that the mouse has an asthma-like condition. Good, thanks for that clarification. I like the participation here. So then, as Dr. Hebert said, when the mice are also infected with this helminth, we see a significant reduction in the eosinophils. So yes, eosinophils help fight parasites, but in this case, the worm is manipulating the immune system in a way that they have less asthma-like condition, because we're looking in the lungs again. So eosinophils in the lungs are not, they're not fighting the worm. This worm lives in the gastrointestinal tract. Eosinophils in the lungs are a sign that this mouse is having asthma-like problems. So this is some evidence showing helminth infection significantly reduces asthma-like symptoms. And the asthma is just a model disease. You can imagine this likely is similar in other inflammatory diseases like Crohn's disease, inflammatory bowel disease, maybe some autoimmune diseases. And I'm not going to go through the data on all those, but there's quite a bit out there if you're interested. So then a similar paper, a similar figure. I won't walk through this one as quickly, or I won't walk through this one as slowly, but IL-4 is a cytokine associated with asthma-like conditions or allergies. And in the lungs, you can see that infection with H. pylori significantly reduces the asthma-promoting cytokine in the lungs. So that's really interesting, right? Now, again, this is in mice, not people, but this is part of the reason there's so much interest in this. Now, what about the microbiome, right? How does the disruption of the microbiome influence H. pylori's impact on airway inflammation? So in this paper, this 2015 paper, they used antibiotics to disrupt the microbiome. So if you use enough antibiotics, you can kill almost everything in the gut and in an organism. Not almost everything, but a lot of it, right? So they're using this to sort of compare what we'd call a control mouse with a mouse that has a dramatically reduced microbiome. And I think I've got audience participation again. So ABX is the antibiotic treatment. HBP is the H. pylori infection again. So the black squares are the worm infection. The open circles have no worm. Okay. HDM sensitization and challenge. So this is the order. This is the order in which these things were done. First, the mice were treated or not treated with antibiotics to destroy their microbiome. Then they were either infected with H. pylori or not infected with H. pylori, because we know the helminths are going to manipulate the microbiome and change their susceptibility to asthma. And then the mice undergo this asthma-inducing protocol, injecting the dust up the nose. So what do you see now? This is just, we'll just say control mice. It means specific pathogen-free. So when you're working with mice in a laboratory, even your control mice, you do regulate the microbes they're exposed to make sure they're not getting sick from, mouse flu and stuff. So what do we see here? Pardon me? Right, exactly. Well, you don't want to get the antibiotic. It seems like this eosinophils, which are a sign of asthma, is worse in the mice that have been treated with antibiotic and had their microbiome disrupted. So that's one point. Although there isn't a statistical test showing you that comparing these per se, but that's suggestive, yeah. And so then what's another conclusion? Exactly right. So without the antibiotic, the helminth dramatically reduces asthma. If the mice have a disrupted microbiome, the helminth doesn't seem to be able to reduce the asthma conditions. So, yeah, I'm going to get to this in a minute, but obviously we're sort of seeing this three-part interaction playing a role. Oh, this is a really interesting study. This is a really interesting figure from the same study, so I do want to share it with you. Is sharing the HPB modified microbiome sufficient to alter airway inflammation? What do I mean by that? I told you that the worm infection changes your microbiome, and we know you can, by being around other organisms in the same species, especially in close contact, like mice living together in a cage, we know that microbiome is exchanged, right? The mice eat their poop, for crying out loud, so eat each other's poop. So they're going to be their microbiomes are going to be intimately connected. So a way to study this is to put mice together. So in this, they've got co-housed mice that have not been infected with HPB, and they do this asthma induction. They look at the sign of asthma by looking at eosinophils and lugs and lots of other things. Then they have mice that have not been infected with the worm, but live in the same cage as mice that are infected with the worm. And it's important to note, because of the timing, it wouldn't be long enough for the non-infected mice to get infected by the worm. But it is enough time for them to eat each other's poop and diversify each other's microbiomes. So what do you see is just living in the same cage as a mouse that's infected with the worm, you have dramatically reduced asthma in those mice, suggesting that the microbiome alone may be sufficient to do this. So really interesting paper. We certainly see three-part interaction here. So the worm H. poli, now I'm kind of homing in on H. poli. That's the worm I studied most of the time during my sabbatical. It infects mice, not humans, so it's a little easier to work with. And it's been used for a long time, so we're pretty good tools for working with it. The gut microbiome we've already talked about, and then the host immune system. So what are some other observations related to this three-part interaction? Well, it's known that earlier I said, could we use things that the worm makes, for example? It is known that the worm makes what we call a TGF-beta mimic to influence the host. TGF-beta is a molecule your immune system makes to help keep your immune system under control. So it's immunosuppressive. And the worm makes a molecule that's very similar to our own TGF-beta, again, to try to hide from our immune system. And it's also shown that this TGF-beta mimic increases these T regulatory cells, which I mentioned earlier are a type of white blood cell that help keep your immune system calmed down. So we certainly, even though this previous study from 2015 kind of suggested it's all about the microbiome, there is plenty of evidence. There is evidence to suggest that H. poli might be acting directly on the host. And I'll come back to that. And then there's other interesting questions as well. Certainly we said the gut microbiome changes during H. poli infection, and we know that changes in the microbiome change the host immune system. Also, there are host factors to consider that I have not yet mentioned, such as genetic differences in mice. Mice of a certain genetic background we call BALB/C, it's just the name of a mouse strain. It's a white mouse, and C57 black 6 is a black mouse. They come from different inbred genetic backgrounds. BALB/C mice are relatively resistant to H. poli infection. C57 black 6 mice are more susceptible to infection. So you see there are host genetic factors that can influence this interaction as well. So how can we tease this? Well, first of all, what are our major interests, me and the Walk Lab and Carlin? What were our big picture questions that we were interested in? What is the effect of the gut microbiome on immunomodulation by H. poli? So certainly that Zeiss et al. 2015 paper we went through a few figures of suggests the microbiome is a major player. But that still leaves open a lot of questions. For example, what members of the gut microbiome are driving that effect? If we could identify, which bacteria in that poop was really allowing the mice to be resistant to the asthma, could we just use that, for example? But the only way to really ask that question is to be able to manipulate the microbiome at a very fine level in mice. And as you guys said, we have tons of microbes all over us. And so how can we have such fine control of the microbes living in and on these mice? Well, that requires what we call a germ-free or gnotobiotic model. So germ-free is basically a mouse that's born, it's a mouse that has no bacteria. It's a mouse living like a bubble boy. You've also heard of like Bubble Boy in like a cartoon or a movie or something. Everything inside its bubble is sterilized. If you're going to put anything in the cage, you have to put it in one of those chambers. You put in gas to disinfect everything in the chamber. Then you open up dual-sided chamber. You're reaching in with those big gloves like you'd see with an astronaut. And the mice are living in there with no microbes at all. What that allows you to do then is add back one at a time or a few at a time bacteria. And that's why we call a gnotobiotic model, meaning there's only certain living things in there in addition to the mouse, not a whole microbiome. So we really wanted to establish a gnotobiotic worm infection model so that they could ask some of these questions. germ-free mouse compared to control mice, and then adding back certain microbes one at a time. Now, we're still not there yet because we're still trying to develop some of the tools in order to get us there. What are the barriers? What are the barriers to accomplishing this? What I was doing was sort of trying to help overcome one of the major barriers to accomplishing this. And that is the fact that these worms come from eggs that are in the mouse poop. Mouse poop is full of bacteria. So how can we get the very first initial worms to be completely free of bacteria and make sure that they're viable, they're infectious, they're able to reproduce? Well, that's basically what I was working on with the help of those other people I mentioned. So how does the H. poly's life cycle work? As I kind of mentioned, eggs are shed in the feces from infected mice. Then what I didn't mention is you have to grow these eggs. Traditionally, you grow them in the mouse poop in a dish. And there's bacteria already present in the mouse poop that provides nutrients that seem to be required for these worms to hatch and then undergo L1 to L2, L2 to L3, undergo 2 molts. So basically morphological or metamorphoses, kind of like a caterpillar changing into a butterfly. But it's a smaller worm changing into a bigger worm. All right. And they have to get to what we call L3. That's the infectious stage for a lot of these worms. And it's known that something from the bacteria is important in allowing those worms to get to the L3 stage, or it's at least suggested. But that was one of our big hurdles. How can we get these worms completely free of bacteria, but they also need the bacteria or something from the bacteria to get to this stage? And if they're not L3s, we're not going to be able to infect mice. So a lot of my time was spent right there, playing with mouse poop, looking for worms, adding bleach to it, looking at the worms growing or not growing, trying to figure out if the worms are growing. Yeah, I don't know. We thought of a lot of things. So this is what it looks like when you're growing the mice in what we call a fecal slurry. So this is just mouse poop with eggs inside of it, and you grow them for about a week, and the little worms hatch out, and we know that they're L3s. We can look at them under the microscope. We can put them in mice. They recapitulate the infection and continue the cycle. Before I got there, Carlin had played with a lot of different conditions to try to get these germ-free worms. And one of them is to use a liquid culture system. Most of the time, when you're growing cells from humans or mice in a dish, what we call cell culture, it's in a plastic dish like this with liquid media, just like sugar and nutrients. So we were trying to use a system like this to grow the worms. And how did that work? Well, you have to use a sugary solution to separate the eggs from the stool. That's kind of a density-based thing. You add this super sugary stuff, and then it helps the eggs separate from all the other stuff in the feces. Then you have to count them. And then we were trying a protocol that used bleach. So we'd expose the eggs to bleach, and part of our process was how much bleach, how long, will the eggs still be viable? Will the worms that hatch from the eggs still be able to make babies? There was a fair bit of testing done before I arrived. So here's some of the work that Carlin did before I arrived, testing different concentrations of bleach. And you can see if the eggs are exposed to a bleach concentration that's too high, like 50% bleach and 50% water, they stop being viable. The eggs don't hatch, they don't survive. So we were mostly working with about a 25% bleach concentration when we're sterilizing the eggs, because you can see that's where viability starts to drop off. And in order to demonstrate that we were really killing the bacteria with this treatment, because even with 25%, you don't know, this means the eggs are still living, but did we get rid of all the bacteria? And one way to do that is to treat bacteria in parallel. So we'd treat bacteria with the different bleach concentrations for different times, and then see, did we kill the bacteria? So pretty basic stuff, really, but necessary in order to get this model going. So you can see some of our example data exposing E. coli to different concentrations of bleach, either in a plate or on a plate, exposing them to different concentrations of bleach for a day or two, and then putting those E. coli either onto a plate, which is what you see here, or into a liquid broth culture. And those are just two different ways to grow the bacteria, because we had to test this a lot of different ways to be sure it was working. And you can see when you're getting up into 15, 25%, the bacteria doesn't seem to survive. So that's confirmation. We were killing the bacteria at a concentration where the eggs seem to be okay. Thumbs up. Now, importantly, E. coli is an aerobic bacterium, meaning it grows in an oxygen-rich environment. Many of the microbes that go in the gut die in an oxygen-rich environment. So we also wanted to test microbes that grow in what we call an anaerobic environment that's completely devoid of oxygen. And you need special equipment for this. And luckily, Dr. Walk's lab does a lot of this. So they have anaerobic chambers that, again, are like those things where you're reaching with the gloves. There's no oxygen inside. Clostridium sporogenes is a species of bacteria that's strictly anaerobic. It will die in the presence of oxygen. We tested to see if we could also kill C. sporogenes with these bleach exposures. And the other important reason is this forms spores, which are highly resistant to things like bleach and temperature. So we wanted to make sure we were getting rid of Clostridium, and it seemed as though we were. So thumbs up again. And this is another variation on that theme we tried, where we had the worms and we added bacteria to the worms to make sure we were killing them. I won't go into that. We also tested to see if we were getting rid of bacteria by looking for DNA from bacteria, because sometimes you might not see them growing on a plate, but maybe there's traces of them that maybe didn't grow. So looking for DNA is a very sensitive way to look for bacteria. And we would see that with these conditions, we could get rid of any signs of bacterial DNA. So that was another sign that it was working. And a third way we looked for bacteria was looking at gram stains. I'm not going to go through all this data, but basically just looking visually under a microscope, do we see any traces of bacteria? And sometimes we still would see traces of bacteria indicated here. But for the most part, we were able to get conditions right around 25% bleach where we got rid of the bacteria also with visual inspection. So 3 different ways, checking to make sure we're getting rid of the bacteria. Then finally, we could put these bacteria, or we could put these, what we think are germ-free worms, in germ-free mice. Or even just in regular mice, just to see, are these worms so messed up from the bleach, maybe they're not going to work at all. And so this is looking at the infectivity of the worms, looking at normal mice. So liquid cultures in gray and yellow, and the traditional method with the worms growing up in the poop is in blue and orange. And it turned out, at least in our early experiments, even though the worms were there, we could see them squiggling around, they seemed to be healthy, they're not really producing much infectivity in the mice. How are we measuring this? You infect the mice, and then you collect the poop periodically and look for eggs in the poop. And that's a sign that the mice are the worms are producing a viable infection. So how many eggs per gram of stool? And we did get eggs produced, and we were really excited about that. So there were some worms in this liquid culture that were healthy enough to reproduce, suggesting there's some potential, but it's still pretty poor. So a lot of our time was, playing with this and trying different conditions to get these worms healthier. I think this is actually in regular mice. This is just in regular mice as a starting point, just to see even in regular mice, can these worms reproduce? We did it both ways. I'm sorry, I can't remember whether this was germ-free mice, and I need to remember to add that to this graph for next time, but I can't remember if this was germ-free mice or regular mice. We did it in both, and we did get similar data in both. But as you can imagine, the germ-free mice are really expensive and hard to come by. So you don't mess with them until you have a little more evidence. So even in regular mice that have bacteria in their gastrointestinal tract, there was problems with these worms. And funny enough, we weren't the only ones working on this. A paper came out while I was there, publishing a modified version of this protocol, and they kind of beat us to the punch. So we learned from that and kind of modified our protocol a little. They were using more of an antibiotic-based treatment. It was a lot more cumbersome and complicated than our procedure. So we're still hoping to polish up our procedure and get it published, because for other people that want to do this, our procedure is a lot easier. And it would make it easier for other scientists to continue this work. So how could we improve it? These were some of the questions we were asking ourselves. If we let the worms incubate longer, maybe they could get more mature. Or what about vitamins? We looked back at some of the papers from the 1960s. They've been studying these worms for a long time and growing them then to see, what sort of things were they adding to the media back then to get the worms to grow. So we looked at what kind of vitamins you find in mouse poop, and then made up different concoctions of liquid media with just those vitamins. Those experiments were relatively unsuccessful. And really, when it came down to it, we could not get the worms to really develop well without bacteria. We still are at a point where we sterilize the eggs with the bleach to get rid of all the bacteria. Then we add in just a little bit of the bacteria we want that's easier to control. We'll let the worms develop and then kill that bacteria with antibiotics. And that seems to be our best case scenario at this point. And there's more I'm not going to go into, but Carlin's still kind of working on that to nail that down. So future experiments, obviously we're trying to get this germ-free model nailed down, and then we could introduce back certain microbes, as I said, to see how they impact this. Two of the really interesting molecules are short-chain fatty acids and kynurenine. Kynurenine is a tryptophan metabolite that is also immunosuppressive. Tryptophan's an amino acid. You may have heard of it, like in turkey. Sometimes people say you get tired from eating turkey. But your body can metabolize, microbes in your body can metabolize tryptophan and make immunosuppressive molecules. So we're interested in the, if helminths are using these pathways at all to be immunosuppressive. So what could I do? One of my goals from sabbatical was to be able to, hopefully continue some part of this research here at Aquinas with Aquinas students. I told Seth that was one of my major goals. So we're talking about maybe applying for a grant, and we've put that on hold, by the way, Steve. That's why I never went to meet with you about that. But applying for a grant to get some tools to maybe be able to genetically modify the worms here at Aquinas with undergraduate students, and then send them back to Dr. Walk's lab, and they can put them in mice, because we're not going to get the mice and all that stuff here. And another question I'm really interested in that I think probably has less people focused on it is, what from the bacteria is allowing the worm to develop into that infectious stage? That's just an interesting biological question in terms of symbiosis, in terms of parasitology, and it could have implications for developing treatments for these worms. Because remember, even though we've been talking about these worms as helpful things most of today, many of these worm infections around the world do cause major problems. So studying these worms could shed other insights as well. Good, so that timing went about well. I guess we're running a little bit long. So big thanks to Aquinas College and all for supporting me as I went away, and especially to my biology colleagues, covering my classes and such. I did get to do some other things. I played in the mountains a lot, which was great. I did some of my first ultramarathons. So this is kind of a joke. Very interesting, unpublished data, never shared with anyone before. I did some shoe testing with running in the mountains. So I compared a really popular new pair of trail shoes, the Hoka Speed Goats, with the Salomon Ultra Glides, and looked at how my heart rate I tried to hold my heart rate steady in a particular zone and repeat that over and over, and then look at the pace that I was running in the miles, and actually found that the salmon ultraglides were about 3% faster than the Hoka Speed Go, which is pretty shocking. 3% is huge. If you're familiar with, is anyone familiar with the Eliud Kipchoge Breaking 2 project? They got a human being to break 2 hours in the marathon with a lot of special techniques. And one of the things they developed is a special shoe for running on the roads called the Nike Vaporfly. And it's reported to make you go about 4% faster. And they originally cost about $280 a pair. So a 3% improvement was pretty exciting. Anyway, so I'll end there, I guess, and ask you if you have any questions. Thanks so much for coming.